on His Majesty's service. Auditory receptors. Hearing, or auditory perception, is the ability to perceive sounds. In the auditory system, sound waves are detected and transduced into nerve impulses that are perceived by the brain. The pinna, or auricle, captures sound waves which enter the external auditory meatus, reach the tympanic membrane, and cause it to vibrate. Its vibrations follow the waveform of the sound. The chain of auditory ossicles transmits vibrations to the oval window where the base and annular ligament of stapes separate the air-filled middle ear from the fluid-filled inner ear. The mechanical vibrations of the stapes create pressure waves in the perilymph of the vestibular duct or scala vestibuli. Through the helicotrema, these waves are transmitted to the perilymph of the tympanic duct or scala tympani, cause the vibrations of the secondary tympanic membrane in the round window, and dissipate. Movement of the stapes in the direction of scala vestibuli displaces the perilymph, causing the secondary tympanic membrane to bulge into the tympanic cavity, and vice versa. The wave motion is transmitted to the endolymph inside cochlear duct or scala media. The pressure waves in the fluids cause the basilar membrane to vibrate. The basilar membrane is wider and more flexible at the apical end and narrower and stiffer at the basal end. Its vibrations always begin at the base and then propagate to the apex. It was shown that the basilar membrane vibrates maximally at different places as a function of the wave frequency. Thus, acoustical stimuli initiate traveling waves which propagate from the base toward the apex of the basilar membrane, growing in amplitude and slowing in velocity until a point of maximum displacement is reached. This point of maximum displacement is determined by the sound frequency. The points responding to high frequencies are at the base of the basilar membrane. The points responding to low frequencies are at the apex, giving rise to a topographical mapping of frequency. In other words, the basilar membrane is tonotopic. It means that each frequency has a characteristic place of resonance along it. The region that vibrates most intensively stimulates the greatest number of hair cells of the spiral organ, which lies on this part of the basilar membrane. The spiral organ, or organ of Corti, consists of epithelial hair cells and supporting cells lying along the basilar membrane and including various types. Pillar cells are arranged in 2 rows, which limit the inner tunnel. All cells which lie between the inner tunnel and stria vascularis are called outer. Cells lying between the inner tunnel and spiral limbus are called inner. Adjacent to the row of the inner pillar cells, there is a row of inner phalangeal cells with a row of the inner limiting cells or inner border cells near it, and epithelium of the inner spiral sulcus further on. Inner hair cells are seated between sides of inner phalangeal cells and inner border cells. Outside the outer pillar cells, there are 3 rows of outer phalangeal cells. Outer hair cells rest on the cup-shaped indentations of outer phalangeal cells. The finger-like processes of the outer phalangeal cells reach the surface of the spiral organ and are located between the outer hair cells. Tight junctions between cells of spiral organ form a barrier that isolates stereocilia and apices of the hair cells from the basolateral parts. Outside the outer phalangeal cells lie 3 to 4 rows of the outer limiting cells, or outer border cells, which gradually pass into the epithelium of the outer spiral sulcus. Supporting cells play diverse roles in the developing and mature spiral organ. They are involved in ribbon synaptogenesis in hair cells. They have rigid cytoskeletons that maintain the structural integrity of the spiral organ during sound stimulation. Supporting cells mediate homeostasis of cations, sodium and potassium, and some small molecules, glutamate and inositol triphosphate, through transporters and gap junctions. Supporting cells also modulate the extracellular matrix by the production of its components. Above the spiral limbus and the spiral organ, the tectorial membrane is located. It is a gel-like structure that exhibits viscoelastic properties. The tallest stereocilia of the outer hair cells make contacts with the tectorial membrane. The inner hair cells have a pear-like shape with a rounded base and narrowed apical part. There are about 3,500 these cells per spiral organ. They have a centrally located nucleus, numerous mitochondria, granular endoplasmic reticulum, Golgi complex, and various proteins. The outer hair cells have a cylindrical shape with a rounded base. There are about 12,000 such cells per spiral organ. They have a nucleus located in the basal part and are also characterized by numerous mitochondria, granular endoplasmic reticulum, Golgi complex, actin and myosin filaments. Motor protein, prestin, is expressed in the plasma membrane on the lateral sides of outer hair cells. It is important for their functions, which will be explained later. Both inner and outer hair cells possess the bundle of 50 to 100 hair-like stereocilia which protrude from the apical surface. Despite their names, they are not true cilia, but large microvilli containing an actin cytoskeleton. Each stereocilium tapers where it inserts into the cell membrane, forming a hinge about which each stereocilium pivots. Actin filaments in stereocilia are located parallel and cross-linked. That's why they are very rigid and do not bend except at the base. The number of actin filaments decreases from hundreds in the shaft to only a few dozen at the base. But the central 10 to 20 filaments are gathered together to form the structure which is called rootlet. It represents the core within the stereocilium, starting halfway down it and extending through the tapered region into the cell soma, anchoring into another structure which is called the cuticular plate. Rootlets contain actin with a higher density of actin filaments than in either the stereocilia or the cuticular plate. The cuticular plate is located below the stereocilium in the apical part of the hair cell. It is a bowl-shaped mesh constructed of cross-linked actin filaments with no directionality. It provides a solid structure that makes a reliable anchoring point for the rootlet. The stereocilia are generally arranged in 3 rows, which are graded in height, becoming taller on the side away from the spiral limbus. The stereocilia in the bundle are coupled by various types of thin filamentous links, which are multiple during embryonal and early postnatal development, but are reduced in the mature stage. Tip links connecting the apex of shorter stereocilium with the side of its taller neighbor are important to consider. They are involved in the mechanoelectrical transduction process. The mechanoelectrical transduction process proceeds as following. When traveling wave makes basilar membrane vibrate at a certain point, the hair cells in this point are displaced. Since the outer hair cells are anchored to the basilar membrane and their stereocilia bundles are in contact with the tectorial membrane, the vertical components of the traveling waves are translated into shear motions between these membranes. As a result, the stereocilia bend at their bases. The stereocilia of the inner hair cells do not reach the tectorial membrane. They bend under the action of endolymph displacements. Within the range of the normal auditory threshold, the hair bundles are displaced about 1 nanometer, and the angle of displacement is about 1 grade. It's worth noting that fluids of the inner ear are different. The endolymph is rich in potassium and poor in sodium ions. It is produced by the cells of stria vascularis. The perilymph resembles other extracellular body fluids, which are rich in sodium and poor in potassium ions. The perilymph leaks through the basilar membrane and fills the inner tunnel and the gaps between the basolateral parts of the spiral organ cells. Here it is usually called cortilymph. But perilymph and endolymph do not mix, because, as it was mentioned earlier, the cells of spiral organ are connected by tight junctions. Besides, in the scala media, the endolymph is about 80 millivolts more positive than the perilymph, which is termed the endocochlear potential. At the same time, the cytoplasm of the hair cells is about 140 millivolts more negative than the endolymph, with the difference between the endocochlear potential and the resting potential of the hair cells being about minus 60 millivolts. This difference in the electric potentials gives rise to the driving force for potassium and calcium ions entry into the hair cells and forms the basis for the mechanoelectrical transduction in them. Being interlinked in the bundle, the stereocilia move together. Deflection of the bundle towards its tallest stereocilium stretches tip links. The increase of tension in tip links leads to the opening of mechanically gated mechanoelectrical transduction channels located at the apices of hair cells. These channels are non-selective cation channels with high permeability to calcium ions. The opening of channels and influx of potassium and calcium ions depolarizes the hair cells. Deflection of the bundle in the direction of its shorter stereocilium leads to closure of mechanoelectrical transduction channels and hyperpolarization of the hair cells. Thus, in response to sinusoidal stimulus, a sinusoidal receptor potential is generated. It may be larger or smaller in proportion to the amount of stimulus and angle of stereociliar displacement. Receptor potentials activate voltage-gated calcium channels at the ribbon synapses, and this leads to a graded release of neurotransmitter glutamate into the synaptic cleft. Glutamate molecules bind to the receptors on the postsynaptic membrane of the cochlear nerve fibers endings and generate action potentials. More glutamate for a longer time produces a higher frequency of action potential and vice versa. One important advantage of mammals is a better sense of hearing, including high sensitivity to quiet sounds. This is due to the cochlear amplifier, which consists in a positive feedback loop within the cochlea that amplifies the traveling wave. Mechanisms that generate force within the cochlea include outer hair cells electromotility and active movements of stereociliary bundles. Outer hair cells electromotility is their ability to convert the receptor potentials into vibrations of their cell bodies. Under conditions of depolarization, molecules of prestin contract and outer hair cells become lower. Under hyperpolarization, these cells elongate. This motor activity leads to a large increase in auditory sensitivity because the outer hair cells increase the amplitude and sharpen the peaks of vibration of the basilar membrane. Active movements of stereociliary bundles have the following pattern. After deflecting the stereocilia a certain distance, a force produced within the stereocilia causes them to deflect further. This force is triggered by the entry of cations through the mechanoelectrical transduction channels. Inner hair cells make synaptic contacts with more than 90% of the afferent nerve fibers of the cochlear nerve. The remaining afferent nerve fibers are in contact with outer hair cells. This is likely afferent component of the reflex arc, which includes the afferent innervation of both the outer hair cells and the postsynaptic parts of the synapses of the inner hair cells. Afferent innervation of inner hair cells is ensured by lateral olivocochlear afferent nerve fibers, which reduces damage to afferent nerve fibers from excessive activation by traumatic sounds. Afferent innervation of outer hair cells is ensured by medial olivocochlear afferent nerve fibers. Their activation leads to hyperpolarization of the outer hair cells, which reduces cochlear amplification. This also protects the cochlea from acoustic overstimulation. In general, nerve fibers supply for inner hair cells is much denser than for outer hair cells. A single inner hair cell is innervated by numerous nerve fibers, whereas a single nerve fiber innervates many outer hair cells. Thank you for attention.